So my name is Mark Schlissel. I'm the Dean of Biological Sciences here at Berkeley, and I want to welcome you all, certainly the people from the Berkeley campus, but more importantly, our visitors from other universities that have come to join us for a day of virology. So today's been organized under the auspices of our Center for Emerging and Neglected Diseases. This is an enterprise we began here on the Berkeley campus and was launched about 3 years ago. And its notion is to provide a common community amongst people studying infectious diseases from a variety of perspectives across the campus, and then now uniting campuses across the Bay Area. It's supported by a generous donation from one of our major philanthropists here at Berkeley, Sam Wheeler. The idea of the center, as I said, is to coalesce work on infectious diseases. There are about 40 or 50 research labs here on the Berkeley campus spread amongst at least 11 different academic departments that contribute to this effort. The umbrella organization, SEND, was the concept promulgated by my predecessor, Jeff Owen, to bring together people doing primarily infectious disease work, but also folks from many departments whose principal focus isn't infectious disease, but they're doing work of relevance. And thus far, it's been quite successful creating community, organizing efforts to attract foundation support, attracting training grants, and then reaching out to other institutions in the Bay Area and also around the world. Virology is a topic that during my scientific childhood, all the best molecular biologists were studying viruses or bacteriophage. It actually gave birth to the revolution in molecular biology. Not that it was forgotten, but it seemed to have shifted out of the limelight for a period of time, and then, of course, underwent an enormous resurgence with the advent of HIV and the discovery of the virus and, of course, of the disease. I don't have to tell this audience its critical importance in the years ahead as both an aspect of great scientific discovery, but maybe more importantly, a major source of advancement in public health. So without further ado, I'm going to introduce Britt Glossinger, who will chair this morning's first session.
So welcome, everyone. I have been instructed to give very short introductions so that we stay on time. And also to remind the speakers, please try and stick to about 25 minutes, leaving 5 minutes or so for questions. But if we run over, I'm going to have to cut the question session short because we have so many speakers today and we don't want to run into lunch or dinner or whatever. So with that said, our first speaker is Professor Jay Levy from the University of California, San Francisco. And he's going to be talking about HIV and AIDS, background, past achievements, and future directions. Welcome, Dr. Levy.
All right. Anyway, thank you. This is a fabulous idea. I want to thank Laurent for bringing virology to the forefront. Those of us, I see friends from long ago, because we're always there to try to get our students and senior people to maintain their dedication to this field, which is so fascinating. Yes, HIV brought it forward, and frankly, I grew up with phage, so it's sort of fun to hear that. I would imagine people who work with phage are wondering, why aren't they talking about lysogeny, things that were really key? And frankly, my introduction to HIV always brings in the idea of latent infections and how one might be able to control this virus that has all sorts of ways of hiding. So I thought I'd begin by putting it in perspective that this is the only talk on HIV, I think, so I'm going to just do a whole broad spectrum of what happened in the early days of HIV and bring it up to the modern time. So just put it in perspective, these are the major epidemics of the world. I'd like to remind my students about this. And we've really only controlled smallpox. We have great vaccines for other of these viruses and these agents, but they haven't worked or they're not being used efficiently. And I find that when I look at epidemics, they go in cycles, and I feel HIV is part of that cycle. And what are the factors that were conducive to the AIDS epidemic emerging? And you see these here, the migration into cities, the poverty, prostitution, the international travel, of course, sexual behavior, the transmission from blood either by IV drug use or intravenous drug use. So I begin with August '81 in the San Francisco Chronicle. These were the 7 deadly symptoms of this new disease that had hit the West Coast. It was also seen in New York and in Atlanta. If you read through this, you'll see it looks just like a flu-like illness, which is why it was so difficult to diagnose. The only thing that I felt was important was a rash, which was a maculopapular rash that was associated with some of the gay men that were coming to the San Francisco hospitals. And at that time, we didn't realize that was really an indication of an acute infection with this virus. And it was only recognized by the diagnosis of AIDS. So that would be pneumocystis carinii pneumonia, which had this agent, which originally was considered a parasite, but then Dan Santee showed that it was actually a fungus. You'd never see this many organism in a sputum as you do in this photo, because it was such a devastating disease for someone who had AIDS, who had what we recognized was a tremendous compromise in their immune system. And we had individuals presenting with Kappa Chisarcoma. I like to point out the linear distribution, almost looking like lymph channels, because I still believe it's the endothelial lining cells of lymph channels that are mostly affected by this. And this later gave rise to the recognition of a herpesvirus HSV8, which is associated with this particular cancer. Hairy leukoplakia, which is the green spans discovered on the surface of the tongue and the buccal mucosa. This is only found in people who are tremendously immunosuppressed and not recognized prior to HIV infection. And then working with Evelyn Lynette here in Berkeley was able to show that it was caused by Epstein-Barr virus, another herpes virus. Essentially, we began to recognize that maybe the side of the tongue was the reservoir for EBV. When you talk about the transmission of this virus by kissing and saliva, maybe that's where it comes from. So here in San Francisco, well, over there in San Francisco, we were hit by this terrible epidemic, and in particular this area here on Polk Street and going down to Castro. And the first person I saw with HIV was a Kappa Chisarcoma patient, Dan Turner, who became a tremendous advocate in the Bay Area. And we began culturing his Kappa Chi cells, and then recognizing there was an immune deficiency, began looking at his white cells. And this is the way the lab was. Essentially, in those days, it was a very small 80 square foot space because the university didn't want us in any other spot. Oh, I will put in a plug. If you're in Washington, part of this lab is now being displayed at the Smithsonian Institute. So I felt really good about it. And what we noticed right away was that there was cytopathic effects. I mean, you do not need a biochemical assay to find this virus. You look at what happens if you filter some fluid from something that shows like this and put it onto fresh peripheral white cells of some unaffected individual, and you see these multinucleated cells, balloon degeneration, unbelievable. When people visit the lab, I show them this. You could do this. You could do this anywhere. And we got Lyndon Oshira here out in Berkeley to take EM pictures of it, and we, at that time, called it the AIDS-associated retrovirus, ARV. You can imagine now, 30 years later, my concern that this is now an abbreviation used for antiretroviral drugs. So I never thought a virus would be caused for a drug. But essentially, leading to the international program and advice, we renamed ARV HIV, human immunodeficiency virus. And at that time, we all voted that wherever your isolate came from, you put as a subscript. So all our isolates have an SF. Unfortunately, most other groups didn't follow this format. So here is the human immunodeficiency virus budding from a CD4 lymphocyte. All those, there's about 1,000 particles there. And because it buds from the cell surface, it carries surface molecules, cellular molecules with it. That's why the early ELISAs were often falsely positive, because if you had autoantibodies that reacted to normal human proteins, they would look like they were positive. So what we didn't recognize in 1981 and 83 when the virus was found was that it had already broken out in Africa. In fact, in 1978 was well recognized there, and in the mid-70s, and became known as the AIDS belt. My own feeling is that this was most prominent and emerged in Zaire, or what is now known as the Belgian Congo. Now, where have we come then since the early days, the early 80s, where we only had one virus, but I must say that our group from the beginning recognized molecularly, and that was working with Paul Lusu, who's here, that there were multiple types of virus, which completely threw the French and, well, more likely the NIH group off, because they thought there was only one virus that would do this, and we showed very early that there was a heterogeneous group. And now you can see that in HIV-1, they have all these clades or subtypes and groups, so the major, the outlier, and the non-MNO, and now a more recent one found in a gorilla, which is a group P. These are molecular classifications in which you're looking at differences of 15% or 30 to 40%. And then it was soon recognized after discovery HIV-1 that there was another agent, HIV-2, which has some distinguishing differences in its accessory proteins. Most importantly, HIV-2 is more so cell-associated, more like HTLV. It doesn't replicate to high levels. You're not going to find it at high levels in the blood. And it doesn't cause AIDS as frequently. And you can go into what we see in the sooty mangami, a very long, persistent situation in which the virus stays within the body and doesn't cause disease. So where are we now with the various groupings? So HIV-1 with 3 major groups, the M group with the 9 clades causing most infections worldwide. HIV-2 has no clades, so all of them are groupings differing by 30 to 40%, and only 2 are really ones that are major in causing problems throughout the world, mostly in Western Africa, passing through India and in China. We have a handful of HIV-2 infections in the United States. I actually have one patient we're following, and it is very difficult to get viral loads on these HIV-2 subjects in the United States. It's just not a subject that's well characterized and well studied. What happens with HIV and retrovirus is because they have 2 RNA molecules that they can recombine. This is an amazing situation, but Rafael Najera gave me, who's in Spain, and that is a recombinant virus of 7 different clades and even an unknown clade. So as we look through this replication of the virus mixing within various continents, you can look forward to more recombinant viruses, and for me, this is going to be one of the big challenges of the future for virologists. So let's look at the worldwide spread. We know the transmission. Those of you, I think, know all this. It's intimate sexual contact, blood, mother to child. There's not any other way of getting this virus. Where is it globally? Up until 2 years ago, when the statistics were available, you're seeing 33 million people infected, and essentially, if you tie it up, close to 60 million people affected by this epidemic. It has 4 million infected this past year, with a person infected every 6 seconds, dying every 9 seconds. Lots of orphans which were never recognized previously. This is the leading infectious disease killer worldwide. In the United States, we have had 1.2 million people infected. I think, just to remind you, that over 50,000 new infections were reported in last year as well. So we're looking at not something that you could just say it's occurring outside of our realm. You're getting 1 to 2 infections in the United States every 15 minutes. Where is it most major? Again, South Africa still is the leading country with the highest rate. You have 40 2 million people living there, so more than 10% of the population is affected. And as you go down through Africa, then through India, and some countries I've labeled, for instance, Ethiopia, which we've had interactions with, they try to hide this, but there's probably over 1 million people infected in Ethiopia. Similarly, in Russia, China still says it's less than 1 million, but it's clearly at least 1 million people infected worldwide. So that this statistic is pretty sobering, that if you are a 15-year-old boy living in a country like Botswana, which is now a little better off than it was when this slide was made, you had a 100% chance of dying of AIDS. And as you go through that, you can see in East Asia as well, like in Cambodia, Burkina. So this is a worldwide epidemic. We have it somewhat controlled. I'm going to show you some of what we're doing now, where we've gotten, but we cannot forget the fact that this has, as an agent that can do just what I mentioned before. It can come from Africa, can become a recombinant there, and come back. And unless we're very, very conscious of this ability of this virus to spread, that we cannot just forget about what's happening in other parts of the world. Again, for the almost 10th straight year, HIV/AIDS is the 4th leading cause of death worldwide. Just thought it'd be fascinating to mention that Wendy Max did these statistics for me in 2008, that you can't say this doesn't have an economic burden, because she estimated that there's $805 billion lost in productivity over the past years from HIV alone. So how does this differ from the other pathogens I mentioned? It directly attacks the immune system, but so does other agents like measles, mumps. It involves, however, incorporation of the virus into the cellular genome. You get a chronic infection, the agent can frequently change, and it can bring other cells into the infection. So for those of you not retrovirologists, and since I'm talking on this, I thought I'd I'd show this animated slide where you have HIV, the double RNA, usually the same, but it can be heterogeneous, two heterologous chains. It will convert into a DNA, and you'll see these, the viral core proteins, the viral envelope protein, and you will see it enter and go to the nucleus. Here are the viral particles. They attach to a particular receptors on the cell surface. The RNA, one single copy, is then copied into DNA. The DNA copies again, looks like a double helix, then enters the nucleus, integrates by integrase into the chromosome of the cell, and then sets up a factory making RNA. Coming to the surface are proteins, and then the virus buds with its envelope from the cell surface. I think you'll see it again. What do we have in terms of antiviral drugs? We can block the attachment, we can block reverse transcription, we can block integration, and we can hit the protease within the particle so that it can't chop the proteins into the correct formation so you get an infectious particle. The receptors are not just CD4, where it was first recognized, but it can be Gal-C in the gut or the vagina, or if you combine an antibody, we showed this years ago, you can get enhancing antibodies, which Eva Harris knows about with the dengue virus, where you get enhancement. You can enter cells through the Fc receptor. So we have a breakdown of viruses depending on, after they attach to CD4, what other receptor they need to get a close binding and then enter the cell. And these are the chemokine co-receptors, receptors that are interacting with chemokines to bring white cells into an inflammatory area. And the virus takes advantage of them. So there are viruses that grow very well in macrophages, which use CCR5, those that grow well in established tumor cell lines that use CXCR4. But there are now about 9 to 13 different chemokine co-receptors. But CCR5 and CXCR4 are the major ones. And you can develop drugs that can block this interaction with CCR5 or CD4 or CXCR4. So why are we seeing this spread of the virus? So the infected people can remain healthy for 10 years at least without showing signs of disease. It can be transmitted by the infected cell. The virus is spread by sexual transmission. We've yet to really conquer sexually transmitted disease. It mutates, and it can become resistant to the immune response. So key, and I must say, if you only go with those not in retroviruses, 2 messages. One, this integrates into the chromosome of the cell. And when it does, the viral infected cell becomes a source of that infection, and it can infect other white cells or mucosal cells. And you see here, this is a macrophage interacting with cervical epithelial cells that aren't directly infectable. But you see all these virus particles are being delivered by that macrophage to the cervical cells. This is David Phillips' picture, and he can show it, this cell marching from one cell to the other, delivering virus. Where's the virus? The virus can be in seminal fluid. And when this virus infects, each time it replicates, you get up to 10 mutations. Now, most of them are lethal, but there is a selection. And when that selection takes place, you can get a virus that is more cytopathic, so it's going to kill cells more easily, or go to various tissues like the brain, the bowel, the gut. And this then gives you a list, and this becomes really important. I'm going to end with, is there a cure possible? You've got to get in early with this virus, depending on its ability, its envelope, can infect a whole bunch of hematopoietic cells, cells of the skin, the brain, the bowel, the kidney, the liver, the prostate, the testes. It can be in a reservoir there, and you need to get rid of it, not just from the immune system. Here's a picture of it in the GI tract, which we showed years ago with working with Jay Nelson. Here it is in the kidney epithelial cells. You're not talking white cells. It can directly infect these tissues, and it can create an infection that becomes almost latent, because you don't get high replication, and just sit there. Very early in the studies, in our group, particularly by Cecilia Cheng Mayer, we showed that if you had an early a virus that came out when a person was healthy, that virus would replicate very low, very slowly, and come out at a very low level. But when that person advanced to disease, the virus came out rapidly and reached high levels. And we then began to see that the early virus, which is now more commonly the R5 virus, doesn't cause the kind of syncytia and cytopathic effects I described for you very early today. And these are more of the X4 type. So what is the hope? Very early in my career in virology, I was taught by the Henleys that look at people who've survived infections. And we did that with HIV, and we found some very early people who were not infected by the virus at all, which was a surprise. They can remain, we call these people long-term survivors. They can be healthy for more than 10 years. We have people 35 years documented infected and are perfectly healthy. Normal CD4 cell counts, low viral loads, low immune activation, and then there's a subgroup recognized now who have undetectable plasma virus. They are still infected, and we have some for more than 10 years. You can't find virus in their blood by a plasma viral load. What is protecting these people? Now, we believe one of the most important responses is innate immunity, where you get cytokines produced, and then that leads to an increase in both adaptive immune response and in innate immune responses like NK cells. And one of the cells that we work on is plasmacytoid dendritic cells, which are the major producers of interferon. And we found very early that if a person reports acutely infected with the virus, if they have naturally a large number of plasmacytoid cells, the interferon-producing cell, they often usually have a low viral load. So that is one protective mechanism. Another we discovered very early in the early 80s from one man, and then have now seen it in many people who are long-term survivors up to the present time. If you put their white cells in culture, you can't get the virus out unless you remove the CD8 cells, and that's shown here, where if you have peripheral white cells and you remove the CD8 cells, you're not seeing any virus, the virus comes out. Now, of course, my immunology colleagues said, well, CD8 cells must be killing. But the fact is you can take infected CD4 cells, add in CD8 cells. You do not kill the CD4 cells. You suppress virus replication. This was an unknown mechanism for CD8 cells. It's now better appreciated in other viral infections. And so you have an ability of the CD8 cell to suppress virus replication without killing the infected cell versus the cytotoxic CD8 that kills. And I'll just mention that Chris Walker and our group showed that if you separated the CD8 cell from the CD4 cell, there is a cytokine made, we call it the CD8 antiviral factor, or CAF, that blocks virus replication. It does it at transcription, and that is now a major emphasis in our lab to define this factor. If a person has AIDS, the CD8 cells large numbers are needed to block virus replication, whereas a healthy individual, one CD8 can control infection in 10 to 15 CD4 cells. Now let me just, and then where is it going? These are the major countries that have been recognized where the future of the epidemic is going to take hold, and they are getting special attention by WHO. What can we do? I like to emphasize the immune mechanisms that one could take. I'm going to just save some time. Immune activation is key. So I'm going to end with these these concepts. Early, we saw AIDS is caused by one virus. Now we know there are at least two. It can infect only CD4 cells. It can infect many cells of the body. Uses only CD4? No, it has other surface molecules. It's a retrovirus? Yes, but it's got accessory genes that make it difficult. It remains stable? Yes, but you can mutate and become more virulent over time. Loss of CD4 cells is by direct cell HIV killing? No, there are other processes, particularly apoptosis and immune activation. No survivors? That was early. No one could survive. That's not true. You have long-term survivors. Adaptive immune response. Those are my immunologic colleagues. I'm sure some of you are here, but this is a virology meeting, so I can talk openly. The innate immune system is also very important. And the adaptive is, as well as the innate, is key. Superinfection, some of you know, people are saying if you can superinfect with HIV, you can't have a vaccine. Let me tell you, as virologists, you know, superinfection is common among viruses. HIV-exposed individuals get infected. That's not true. I didn't get to talk to you about there are people who have exposed many times. They're not infected. Will an AIDS vaccine become available? Key. We've never had a vaccine that prevents infection with the infected cell. So here are some of the characteristics of what we're looking for with an ideal HIV vaccine. These are the problems we face. This is, okay. I hope this is in here, but if not, okay. I did want to say one thing, that there's a major emphasis on cure, and the Berlin patient, the famous person who was transplanted with a stem cell which lacks CCR5, is doing extremely well 4 years later, curative, well, sort of cured from his leukemia, functionally cured of HIV. This is a new direction. Our group is looking at it as well. Think about it. If you could replace the immune system with cells that lack CCR5, you would have a major impact on preventing the virus from taking hold, and that immune system could then control the infection, as we see in long-term survivors. And these are the approaches that I think are going to lead to the best impact, so we don't have to use antiviral drugs, and we can get to approaches that are more natural. So thank you all very much.
I think we have time for one or two questions, and there should be some mic runners around, so.
Yeah. So let me say what the idea is with this stem cell. You would take then stem cells from an infected person. You would manipulate them so they lack CCR5, and then they differentiate into all the white cells of the body. They would then be resistant to the major subtype that's found early in infection, and that's the R5 virus. Now, what the Berlin patient told us is that even that person had 3% of his virus was X4. So it should be, that virus should be able to take hold. And we believe that the natural immune system, we're pretty sure the CD8 cells are doing something to prevent the infection by the X4 virus, which I'm convinced is still in his body. You saw all the cells infected. There's no way that Giorgio Hutter, who did these studies, can go in and check the brain, the testes, the bowel, all these. They tried the bowel to see there's no virus present. So that is, I think, really the most forward way of looking at trying to bring a functional cure in this. Yeah, Paul.
Just a comment. As you know, many animal models now, feline immunodeficiency virus, SIV, that can help us investigate molecular mechanisms of pathogenesis and all that. And I know in the interest of time, you didn't have the chance to.
No, Janet Yamamoto was just yesterday at our lab. She's looking at a tremendous approach to the vaccine. I'll just share it with you. She found by chance, someone called her from the clinic and said, you know, this man shows ELISA positive for HIV, and he's no risk. He shouldn't be infected with HIV. So she said, does he have a cat? Yes, he has a cat. There's a cat. The cat has FIV. And they went and found that he has responses to FIV. And then Janet found there was a cross-reaction of FIV to HIV. So now she's working with us, and we're finding that a lot of our long-term survivors have response to FIV. So guess where it's going? Make an FIV vaccine, and you've got control of HIV. Anyway, thank you all very much.